Hey there, guys. So I'm Cassandra Swett, and this is a proposal that Amanda Hodson and I are co-submitting to look at the interaction between root knot nematodes and diseases and to improve diagnostic tools for resistance breaking root knots. So Amanda is going to tell you more about the resistance breaking root knot situation. You're up.
Okay, so we've had kind of increasing reports of root knot nematodes on cultivars that have the MI gene, which is supposed to confer resistance to root knot nematodes. And there's several possible explanations for this. One is that the root knot nematodes could be mutating so that they're able to overcome the resistance in the tomato. It's also possible that there's some kind of stress like heat or another disease that is allowing the nematode to infect the plant because the MI gene has not functioned properly. It's also possible that there's other nematode species present that are potentially not sensitive to the MI gene at all. This could include Meloidogyne hapla or potential invasive species like Meloidogyne floridensis. So Antoon Plug did a previous study funded by CTRI, and he found that 14 out of the 16 isolates he collected were resistance breaking nematodes. And these were on resistant cultivars, and 2 of these were not. And this highlights the fact that there might be some sort of stress thing going on where it's not just a genetic effect. So of the 14 isolates he found, only 5 of these were ID'd to species. 4 of them were Meloidogyne incognita and 1 was Meloidogyne havanica. Next slide. Great. So what this highlights is that we really need to understand how biotic and abiotic stresses are interacting to influence nematode resistance. And this will allow us to develop better management strategies. We need better ways to figure out if we have resistance breaking nematodes or not, so that we can have better decision support. Because if these nematodes are resistance breaking, then changing the cultivar is not going to help because they will all have the same MI gene, and chemical management is the only option. And problematically, there's no rapid diagnostics right now to tell if you have normal or resistance breaking populations. It requires an 8-week greenhouse bioassay. So that brings us to the next slide, developing molecular-based methods to detect resistance breaking root knot nematodes. So our first sub-objective is to establish statewide data on the incidence of root knot nematodes in fields that have cultivars with the MI gene. And we will confirm whether these populations are normal or resistance breaking by growing them on resistant cultivars in the greenhouse for 8 weeks. And then, since we want to know what species we have, we will sequence the isolates to identify who they are. This was not done previously in the other project. And the second sub-objective, we will examine the sequences to see what kind of variability they have and compare the variability between normal and resistance breaking populations. And Antoon has generously agreed to let us have his 10 isolates from the previous project, so we'll be sequencing those as well. Next slide. So some of our deliverables for objective 1 is that we will have determined the current extent of resistance breaking root knot problems. And in contrast to the previous study, we will have got a species ID for all of the resistance breaking nematodes we find. At these sites, we will also have characterized what other cofactors are occurring that might predispose the nematodes to become resistance breaking, including abiotic and biotic stresses. And then we will have identified preliminary diagnostic regions that could be used as biomarkers or for future diagnostics. And with that, I will turn it over to Cassandra.
Yeah, so as Amanda alluded to, although root knot nematode and resistance breaking is a huge problem across the state in and of itself, there's actually a lot more going on with this problem. And so one of the things that we're learning, and that many of you already know, is that there are a lot of fields that have both root knot, nematode, and one or more other diseases. This is showing an example of a field that I went to earlier this year that has these big patches you can see that was root knot nematode, but also had Fusarium wilt. And the issue here is that you can get both diseases and root knot nematode interacting, causing issues that are much more severe than either alone or even, what we say, additively. So across the state, I've seen this dozens of times. Root knot nematode is co-occurring not just with Fusarium wilt, but with other Fusarium diseases like the crown rotters, southern blight, as well as bacterial canker and viruses. And we don't really know what it could be interacting with to be causing these complex effects. So we want to examine these disease complexes and what is involved in these complexes. And we also more particularly want to start to really look at that Fusarium wilt root knot interaction. And one of the things that we're particularly concerned about is the potential for root knot to compromise Fusarium wilt resistance in F3 cultivars. And this is something that we have seen in the field, F3 fields with Fusarium wilt that have severe root knot. And there is data to suggest the root knot can compromise resistance to Fusarium wilt in the case of race 1. So we have proposed a pretty simple field study that I think can generate some really good data on this. We have a total of 6 grower collaborators that we have identified that have both root knot nematode and at least one other disease problem. And they've all agreed to participate in this study in collaboration with ourselves, as well as Tom Cherini, Brenny Agater, and Amber Vincesi Ball. And what we're going to do in each of these fields is just apply nematicide in part of the field and not apply nematicide in the other part of the field, and see if the nematicide reduces both root knot nematode, which it should, and the other disease or diseases that are occurring in that field. And this would tell us what might be influenced by root knot, as well as the potential for nematicides to control these complexes. So we'll be looking at diseases, characterizing those diseases, enumerating their impacts in the field, and looking at yield. And then to further establish whether root knot could be compromising Fusarium wilt resistance in F3 cultivars, we're proposing that at least one of these commercial fields where growers already confirmed they will grow an F3 cultivar and hopefully more, we're going to evaluate particularly whether Fusarium wilt develops in that F3 cultivar in the absence of a nematicide, AKA when root knot is present and affecting both plants. And then simultaneously to look at this in a more controlled situation that removes field variability, doing the same trial in a greenhouse, but with more F3 cultivars, and looking at co-infections with root knot and Fusarium wilt versus just Fusarium wilt alone. So our deliverables for this, I think, are going to be pretty impactful for a single year study. Look, we're going to be able to say what are the disease complex players. Are nematicides going to help control these disease complexes? Is there an effective root knot on Fusarium wilt resistance that we need to be managing? And then I think this is a long-term project. Amanda and I both feel that there's a lot more to be done. And we would really like to use this preliminary data for multi-year project proposals to state and federal programs. So this is the breakdown of our budget as detailed in the proposal for a total of $35,298 divided between Amanda, myself, and the farm advisors. And the budget for Amanda and I is predominantly to do all this laboratory work, looking at root knot nematode resistance and diagnosing diseases, as well as the field work that we're doing, the farm advisors supporting the field work that they're doing. So thank you so much for listening. And I hope that you have some questions and would look forward to your contributions and insights into this project. So hopefully we'll talk to you later.